Hi, my name is Claudia Zhang and my project is on the use of proteomics and bioinformatics to design cancer vaccines. Cancer is a growing issue, with 1 in 6 deaths being caused by cancer, and that number is on the rise. So, what exactly is cancer? Cancer is when your own body cells mutate and suppress the immune system to avoid detection. Traditional methods of cancer detection involve invasive and costly procedures such as imaging and biopsies. This study will focus on a more non-invasive and personalized method of detection that uses immune signatures and blood biomarkers. First, a bit of background. What does a viable vaccine need to do? Well, it needs to be able to stimulate both parts of the immune response, the innate phase and the adaptive phase. The innate phase response involves the antigen-presenting cells, APCs. These cells take an epitope, piece of foreign molecule, and present it to more specialized T cells. If the T cells aren't stimulated by APCs, the rest of the immune response does not occur. The stimulation of T cells initiates the adaptive phase response. The T cells are specialized killers, and they later keep memory of the foreign body. However, it is important to note that if presented with an epitope, the T cells may or may not respond depending on whether they are suppressed or inflamed. An essential part of vaccine design is manipulating this suppressed or inflamed response. This study explores 2 big questions. How is the immune system being suppressed? And can we use bioinformatics to identify an epitope that can reactivate the immune system? First, we'll address how the immune system is being suppressed. Tumor cells produce 2 types of proteins, soluble secreted proteins and membrane-bound proteins. We want to know if both types suppress the immune system, and which part of the immune system do they affect? Innate, adaptive, or both? This experiment used the following cell trials: THP1 cells for the innate response, peripheral blood mononuclear cells, PBMCs, for the adaptive, 4 cancerous lung cell lines, and 1 healthy cell line as a control. The lung cells were grown, and as they grew, they produced and secreted their proteins into the supernatant. This supernatant was then collected, and the THP1 and PBMCs were exposed to these cancerous proteins. A multiplexed proteomics assay, the Luminex assay, was then used to analyze the immune response. The immune response is measured by the concentration of different cytokines, chemicals, it excretes. Some cytokines are inflammatory, so they encourage the immune response, while anti-inflammatory cytokines suppress the immune response. In the innate phase response, we are looking for an increase in the inflammatory cytokines GM-CSF, IL-1 beta, IL-6, IL-8, and MCP-1. When THP1 APCs were exposed to soluble cancer proteins, there was a significant increase in inflammatory cytokines compared to the control. Thus, the innate phase response is functioning properly when it comes to soluble cancer proteins. On the other hand, when the PBMCs, T cells, were exposed to these soluble proteins, there was a decrease in inflammatory cytokines, IFN gamma, in PBMCs treated with A549 and H441 soluble secreted cancer proteins. Thus, the T cell response is suppressed in certain cases by soluble cancer proteins. Now we do the same experiment again, but with cancer cell lysates, which are membrane-bound proteins. Again, the THP1 cells were inflamed, so the innate APC response is functioning properly. However, we again see a decrease in inflammatory cytokines in PBMCs treated with A549 lysed membrane-bound cancer proteins. Thus, the T cell response is suppressed in certain cases by membrane-bound cancer proteins. So to answer our first question, how is the immune system being suppressed? Well, the adaptive phase response, T cells, is being suppressed to varying degrees by both soluble secreted and membrane-bound proteins. Next, we want to know, can we use bioinformatics to identify an epitope that can reactivate the immune system? In essence, can we create a vaccine with bioinformatics? Traditionally, we designed vaccines by sequencing and testing the entire foreign body's genome using in vitro assays and other costly methods. We're really just testing each chunk of the genome and eliminating options by guess and check. This is why traditional vaccine design can take so long. This study uses algorithms, machine learning, and bioinformatics, which allows for greater efficiency and accuracy. Remember how I mentioned earlier that epitopes must be presented by an APC to start the immune response? Well, that's why an epitope's affinity for binding or interacting with an APC is so important. Epitopes bind to major histocompatibility complexes, MHCs. The better the binding ability, the stronger the immune response, the better the vaccine. There are two categories of MHCs, MHC class 1 and MHC class 2, but we'll focus on MHC class 2 since those are more specialized. For humans, there are 6 different MHC2 alleles possible, but each individual person really only has 2 alleles. As such, people with different MHC alleles will respond differently to the same epitope. So here you can see a model of epitope binding. Think of the epitope and MHC as puzzle pieces. You want a snug fit for strong interaction and good response. Now, the computer can run thousands of these models and rank them to show which epitopes bind the best. The higher the z-score, the better the binder. So in essence, the z-score ranks each epitope's effectiveness. Now, there are 2 paths to go down depending on what kind of vaccine you want. A personalized vaccine, where you need an epitope that is very effective for an individual's 2 MHCs, or a widespread population vaccine that needs an epitope that is broadly effective for all 6 types of MHCs. Here's another excellent visual that in silico methods can produce that allows researchers to easily access information and determine a vaccine's effectiveness. In my study, I used the Immune Epitope Database, IEDB, and the consensus algorithms to determine a high binder from the lung cells. Here you can see the first epitope is quite promising. It binds to multiple MHCs. Similar epitopes have been tested in other research groups, and its properties may be similar to another known protein. Now, this epitope can be tested in vitro assays to verify its binding affinity and predicted interactions with cells. Bioinformatics has allowed us to bypass the brute force guess and check method of traditional vaccine design, saving us both time and money. So in a nutshell, yes, bioinformatics can quickly and effectively predict potential epitopes as well as their behavior in vitro and in vivo assays. This study concludes that 1. Tumor proteins can modulate the immune response to varying degrees. 2. Comparing the cytokine profile, immune signatures, of cells exposed to cancer proteins to healthy baseline profiles can non-invasively detect and monitor cancer. And 3. Algorithm-driven design of an antigenic cancer epitope would be able to boost the inflammatory immune response and allow the immune system to detect cancer. Although this experiment successfully proved that bioinformatics can lead to a more streamlined and informed cancer detection and vaccine design, it is only a proof of concept. Future studies could test methods of vaccine delivery, compile a database of genetic and proteomic profiles, and customize the process by delivering the neoantigens from the tumors associated with the subject.